with microglia and PTSD and neuroautoimmune disorders. So back in 2019, 2020, during, of course, COVID, everybody was in home. And I was working on a theory of PTSD, specifically it being an autoimmune disorder based off of the functioning of microglia. So to backtrack a bit on what microglia are and how all of this kind of fits together. So microglia, to put it really simply, I'm pretty sure all of you all are astute in this anyway, but to put it quite simply, they are like the white blood cells of the brain in the sense of they mediate inflammation and they help address disease and misfiring in the brain. And so just like in our body, sometimes our immune system may get overactive and we have what is an autoimmune disorder, like, maybe lupus. In the brain, the same thing can happen. Microglia can get really excited because of something happening, and that could be a chemical response. And as we will talk about a little later, could also be an environmental factor. And when microglia, in their normal functioning, they will eat synapses or trigger a process that reduces inflammation. When they get overactive, they get a little bit too hungry and they can cause deterioration or gray matter atrophy is what it's called. There have been studies done in recent times, but about a decade or so ago, where this was already evidenced in psychological disorders such as depression and schizophrenia, where there is an atrophy in certain areas of the brain in gray matter. So prior to 2019, 2020, I did a study on glucose metabolism and gray matter atrophy in different areas of the brain and how it impacts those areas, specifically in relation to PTSD and chronic pain. And after I was finished with this study, I noticed that the budding studies that looked at SPECT MRI scans of PTSD were very similar to the other studies that other people had done on depression and schizophrenia. So I was like, is this kind of the same thing? So there was a lot of parallel thinking going on in the field and the world, which I think is beautiful because other people also had this idea. And at the same time that I'm working on my theory, there were studies that were going on that we are just now finding out about that have been published in recent years. So 2021 is still being studied because we're right on the cutting edge of what this phenomena is or may be. And so the long and short of the microglia becoming overactive and having this inflammation response is what we are basically gathering from this as of now is that PTSD is potentially a neuroautoimmune disorder, which is very different than our conceptualization of PTSD now. We know that there are neuro effects of PTSD, but if this is a neuroautoimmune disorder, then it's the first autoimmune disorder that we know of that would be strictly caused by the environment. And it transforms how we would treat it. And so just a little bit about me. I am a clinical psychologist. I specialize in trauma as well as neuro. And so what I specialize is more of ASD, and so autism spectrum disorder and ADHD as far as neuro. What I have been kind of moving toward is PTSD and neuro. And so I say all that to say, when you are a hammer, everything is a nail, and that's my hammer. So when I was looking at these studies of depression and PTSD, I was thinking more of, in my field, of mass trauma, which is what I specifically study. And so mass trauma is if you think of veterans who have been combat deployed, that of course is a trauma that we often think of, but other mass traumas could be genocides, which I've researched and studied, as well as, you know, natural disasters. What we may not think of as mass trauma, but actually is mass trauma, is also the systemic being disenfranchised, and as far as people of color and minorities, and how that is also a very wide trauma, at least here in America and in other countries as well. So that is also a form of mass trauma that has gone on for generations and decades. To bring this together, these complex traumas that are sustained over a long period of time, if they induce an autoimmune response, how do we merge the treatment of what we see every day? How do we merge the medical and psychological aspects to create a blended and more comprehensive approach to treatment? The same that we do with a lot of mental health disorders, so schizophrenia and depression and a lot of anxiety, a lot of disorders that we are already doing that quite well. How do we use that for PTSD, keeping in mind that it's very much environmentally based? And so there are some studies that have recently, again, we're just on the edge of this, so there's nothing that's well researched or evidence-based as of right now. They're still in the lab doing studies. And so there was one study, it was on mice, and they did a single prolonged stressor. And then they looked at the microglial activation. And the alterations, it was a suppression of microglial activity is what they found. It lasted between 1 and 6 days. On the 7th day, there was no noticeable change. And so we are seeing that, I have some questions. Well, let me finish my sentence and then I'll address those, but thank you for asking. And so what I lost my train of thought, I'm sorry. Okay, and so at that point, off of a single long-term stressor, a single prolonged stressor, I'm sorry, there were no effects after 7 days. And so they are saying that this can, at least for a single stressor, a microglial overactivation can reduce, and there can be neuroregeneration, and therefore the effect can be treated over time. And studies they have done in humans, again, these are just studies, but they have found transmagnetic stimulation to cause microglial suppression and neuroregeneration, as well as foot shock therapy, so a form of electrotherapy is helpful as well. What I would really like, practitioners, those of us that are practitioners, to just think about is how we conceptualize the people that we see every day, And how do we look at PTSD from more of a blend of a medical and psychological model, the same way that, you know, if we're treating depression and someone who is severely depressed, we often say it's a hand in hand of medical as well as psychological treatment. And and so I'm sorry, there's more questions that are popping up. And so if we can have that same philosophy for, you know, kids that are under prolonged stress, or a person who goes under, or an adult who has had significant trauma, how would that alter our current understanding? I'm going to hop to some questions now because that's quite a list. And so what is the role of microglia in TBI? What I'm talking about is PTSD, not TBI. And so PTSD is, well, I'm sure you know what PTSD is, but it's exposure to a life-threatening event. A TBI is an actual brain injury. It's a physical, to some form of brain damage. And so those would be a bit separate. So to answer how the role of microglia affect TBI specifically is out of my wheelhouse, unfortunately. How does stress affect microglia and PTSD? And so basically, the mechanisms that are, and Bob, let me pull it up. But the long and short of it is that, so I'm pulling up the my actual study to look at how it, the specific mechanisms. I'm assuming that's what you are wanting, is like what proteins are involved in all of that. Is that what you are looking for? Hold on, I lost the tab where we are. Research stress results in acute and long-term change. Yes, that's correct. And so it's the hippocampus, it's the amygdala. Actually, it's a whole brain impact. I'm looking at the comment. I will read it out loud. How does stress affect microglia and PTSD? What is the role of microglia in TBI? I've addressed that. What triggers microglial activation? It's stressors in the environment, and it does cause a chemical activation, but it's a whole brain. And that was what I found in the study before that I was referencing when I looked at gray matter atrophy and glucose metabolism. It wasn't in regard to microglia in that specific study. The theory on microglia came after, but it's a whole brain effect. And so the temporal lobe, like it's, there are chemical changes in the glucose metabolism in almost every part of the brain. There isn't gray matter atrophy in every part of the brain. Specifically, the hippocampus and the amygdala have the most gray matter atrophy. And so as far as the chemical changes and the brain regions involved being the amygdala, the medial prefrontal cortex, and hippocampus, yes, it's those, but it's also others. What are the neurobiological systems involved in PTSD? What do you mean? They're all involved. They're all involved in some way. So when you say neurobiological systems, help me out in clarifying what you are specifically asking, Dr. Dahal. But that's all that I have. I'm here to answer any questions, any feedback, but that's all I have. We aren't going to go on for too long. I'm trying to see if there are any more. I guess I'm waiting on a follow-up to Dr. Dahl's question. Maybe there's no follow-up. So that's all that I have.
All right. Thank you so much, Dr. Lankster, for explaining the PTSD autoimmune nature.
Thank you.
Yeah, you already took so many questions in between. That was impressive.
No problem. Well, I thank you guys, and I guess I'm gonna hop off.
So let me ask if anyone else has any questions for Dr. Lankster. Yeah. So dear attendees and other speakers, if you are having any other questions, please come up and ask your questions. Raise your hands and ask your questions. Unmute yourself. And thank you so much for the chain of questions, Dr. Dahal. I think it was quite informative for everyone that they got some add-ons also. Thank you so much. Yeah, thank you, Dr. Lankster, for taking all the questions.
No problem.
So, time for our next speaker. So, let me ask Dr. Tshetiz Dahal to come up on the screen and yeah, let me introduce till then. Everyone, next speaker, our second speaker is Dr. Tshetiz Dahal from Ukraine, and the title of his talk will be Beta-Amyloid Peptide and the Alzheimer's Disease.
Thank you, Ghazal.
Welcome, doctor. Good evening to you.
Good evening. Can you see my screen?
Yes, perfectly.
Okay. So shall I start?
Yeah, screen is all yours. All the very best.
Thank you. So welcome everyone to International Life Conference organized by OLCAS, Memory Disorder and Conditions. So for today, my research title is Beta-Amyloid, Peptide and Alzheimer Disease. So hence it is published in two reputed journals, J Neuroscience, Journal of Neurology and Neuroscience, and iOS Journal of Dental and Medical Sciences, cited by around 1,200 people all around the world. I am myself Dr. Tshetiz Dahal. I am general physician, clinical researcher, and writer. And I am from Lugansk State Medical University, Luhansk Oblast, Ukraine. But I belong to India. So hence, first, we need to understand what is Alzheimer disease. So Alzheimer disease is the most common type of dementia. So it is a progressive disease beginning with mild memory loss and possibly leading to loss of ability to carry on a conversation and respond to environment. So Alzheimer disease involves part of brain that control thought, memory, and language. So I'm quickly moving to my abstract of the research. The formation and deposition of amyloid peptide, that is A, are largely thought to be responsible for development of Alzheimer's disease. So researchers like me have been perplexed by sluggish to non-existent association between the degree of clinical dementia and the amount of neurotic plaque disease in human brain for a long time. So this question has been resolved, and thanks to recent developments in our knowledge of origins of amyloid disease. So the solubility of amyloid and amount of an amyloid in various pools may now be more directly tied to disease state according to substantial evidence. So the makeup of these pools of amyloid reflects various populations of amyloid deposits and is clearly correlated with the patient's clinical condition. So using imaging methods such as novel amyloid imaging agents based on chemical makeup of histology, dyes, it is now possible to monitor the development of disease in a living patient while also monitoring the amyloid pathology. So it's interesting that these methods show that amyloid deposited in AD differs from that discovered in animal models. It's different in human models. But in general, deposited amyloid does not exhibit the same physical and biochemical properties as the amyloid observed in Alzheimer's disease and is more readily removed from the brain in animal studies. So important questions about the creation and evaluation of potential medicinal agents are also brought up by this research. So you can see in the figure, like just about the neuron in the normal brain, it's shown, and the neurofibrillary tangles in opposite, it's attacked by the amyloid plaques. So you can see in the figure, the amyloid plaques are getting back to the neurons. So this is how the Alzheimer's disease is attacking the neurons and you're getting Alzheimer's disease. So this is what the difference between amyloid plaque and Alzheimer's disease with the normal brain. So we need to understand what are amyloid plaques. Firstly, to know the research. Amyloid plaques are a disorder that are accumulation of beta amyloid proteins that come together between the nerve cells, neurons in the brain of Alzheimer's disease patients. So why does amyloid beta cause Alzheimer's? So research have shown how a protein fragment known as beta amyloid, strongly implicated in Alzheimer's disease, begins destroying synapses before it clumps into plaques that lead to nerve cell death. So what increases beta amyloid? Increases in metal ion accumulation and oxidative stress in AD brain are associated with changes in concentration of soluble and deposited amyloid beta. So it has been shown that metabolic stress promotes ABPP expression and changes the metabolism of A beta protein from non-amyloidogenic to amyloidogenic pathway. So where is amyloid beta protein found? Amyloid beta is produced in the brain throughout life, and it accumulates in the cerebral cortex in elderly and to an excessive degree in Alzheimer's disease. So where is amyloid beta found? Beta amyloid is present in brain interstitial fluid, that is also known as ISF, and is considered a metabolic waste product. Mechanisms by which amyloid beta is cleared from the brain are not completely understood still, although there is evidence that sleep plays an important role in amyloid beta clearance. So we are moving to the introduction. The extracellular plaque deposits of the amyloid beta and the flame-shaped neurofibrillary tangles of microtubule binding protein, tau, are the two hallmark diseases necessary for a diagnosis of Alzheimer's disease. Mutations in preservanin 1, that is also PS1, preservanin 2, these are the proteins, PS2, or amyloid precursor protein, APP, which is the precursor protein for amyloid, are linked to familiar early onset types of Alzheimer's disease, which is also mainly known by preservanin 2, that is also a protein for Alzheimer's disease. So the catalytic subunit of secretase, the last endoprotease in the pathway that produces the peptide, and can be either PS1 or PS2. The amounts and distribution of amyloid beta deposition are only weakly correlated with the clinical manifestation of the disease. Despite this genetic evidence and the demonstrated involvement of amyloid beta in inducing synaptic dysfunction, disrupting neural connectivity, and association with neuronal death in a brain region specific manner. So clinically, Alzheimer's disease is indicated by slow and steady decline in cognitive function, and neuropathologically, the condition is indicated by the presence of neurophil threats, loss of specific neurons, and loss of synapses, in addition to the classic signs of neurofibrillary tangles and senile plaques. So for the benefit of doctors and medical professionals, the techniques for evaluating the pathology and categorizing the stages of Alzheimer's disease have been standardized and codified. So the density of tau protein and neurofibrillary tangles and neuritic amyloid plaques in the affected brain area is the standard indicator of pathology. So the presence of neuritic plaques composed in large part of highly, that is insoluble, amyloid beta in the brain parenchyma is required for a diagnosis of Alzheimer's disease. So the deposits of tau protein are also present, although they are also found in number of less common neurodegenerative disease, notably in absence of neuritic plaques, not amyloid plaque, but in neuritic plaques. So the neurofibrillary tangles in different diseases have some distinctive morphological features and may exhibit a distinct composition of tau isoforms that differs from Alzheimer's disease. So the process of creating and has not been straightforward. And existing systems are not entirely accepted, but the final decision about the staging of disease state is still made by clinicians, neuropsychologists, and pathologists in clinico-pathological ways. So the staging system is significant drawback is that is just can be only be applied roughly to living subjects. Even though we try with the animal subject, but we can mainly diagnose with only with the living subject. So it is necessary to use a clinical diagnosis of probable Alzheimer's disease because the pathology of AD is only discovered at autopsies. So research on this disease causes is severely impacted by the absence of an in-life diagnostic test, which is especially problematic for clinical trials because it adds further heterogeneity to the subject population. So in particular, if the stage of disease is ambiguous or the patient population is not well characterized, therapeutics cannot be fully tested if they must be delivered before the disease is advanced past a certain stage. So amyloid beta deposition is only tangentially connected to the severity of dementia, has long perplexed researchers studying Alzheimer's disease. So the amount of amyloid beta deposition may have potential effect that could contribute, but it's also possible that amyloid beta exerts its main effect sooner by starting a chain of events that once started continue with amyloid beta. But the human amyloid beta vaccination trial may provide some evidence to back up this claim, and there was a vaccination called AN1792. So the brain amyloid beta deposition in these cases, excuse me, the brain amyloid beta deposition in these cases was far lower than might be anticipated based on historical levels for a particular clinical stage. So despite this noticeably smaller level of amyloid beta, which is probably brought out by immunotherapy, the participant cognitive loss persisted to the point of end-stage dementia, which is clinically identical to untreated Alzheimer's disease. Since we have no way of knowing the pre-treatment amyloid load and the number of instances is too low for a true cross-sectional comparison, this is not absolute proof that the elimination of amyloid beta was successful. But it is allowing to assume that these findings indicate that amyloid beta work as a catalyst for a degenerative process that persists even after it is withdrawn. So although hence we have done an experiment in animal brain model, the exact mechanism is unknown. So this is a challenging theory to verify, though as it calls for accurate diagnosis of Alzheimer's disease in patients at a very early preclinical stage. But at a field that is now impossible even with the most accurate and sensitive method of disease diagnosis. But another explanation is that the disease accompanying enormous neuronal loss is caused by a particular form, but or a kind of amyloid beta. The ability of technique used to quantify amyloid beta differentiate between disease-related AB and less important form that weakens the link with clinical stage. So in prion illnesses, where the same protein sequence can take on various disease, causing conformation, each of which causes neurodegeneration in a specific distribution of brain region and yields a different clinical appearance, we find a parallel to this situation. As you can see in the figure below, the Alzheimer's disease of a mouse brain in the right side and the L1 treated mouse brain in the right side of amyloid plaque with activated microglia and p-tau protein that which is aggregating the situation. So in this research, we propose that Alzheimer disease in amyloid beta is polymorphic. So producing conformational form a distinct pool of amyloid beta, some of which are more relevant to disease than others. So method and system to identify these pertinent types are improving, which will allow this theory to be tested. So moving to the catabolism and clearance. So the primary component of amyloid plaques in the brain and cerebrovasculature of Alzheimer disease and Down syndrome. Patient was first identified as below 4 kDa amyloid beta peptide, which was produced from bigger APP. So although the exact role of APP has not been determined, substantial study has enhanced our understanding of how the amyloid beta peptide is synthesized and then either transported outside the brain or destroyed there. So the interaction of these elements determines the final amount of amyloid beta that forms amyloid plaques in the brain. So the age of disease, onset and disease duration may change as the disease progresses. The enzymatic process responsible for metabolism of APP to amyloid beta are now reasonably well understood. But APP is sequentially cleaved by two membrane-bound endoprotease activities, that is beta and gamma secretase. But beta secretase first cleaves. So APP to release a large secreted derivative, hence also known as APP beta, but a fragment of 99 amino acids, that is also CTF beta, which begins with N-terminal aspartyl residue of amyloid beta, but remains membrane bound and is in turn rapidly cleaved by gamma secretase to generate amyloid beta. But cleavage by gamma secretase is somewhat imprecise, so resulting in a C-terminal heterogeneity of resulting peptide population. Hence, numerous different amyloid beta species exist, but those ending at position 40. So amyloid beta 40, hence we talked about 99 amino acids. So amyloid beta 40 are the most abundant, minus 80 to 90%, followed by 42, AB 42 to minus 5 to 10%. The slightly longer forms of amyloid beta, particularly A beta 42, are more hydrophobic and fibrillogenic and are principal species deposited in the brain. So beta secretase activity is believed to be the rate limiting step in amylogenic pathway and process 10% of total cellular APP. So hence CTF alpha produces the more benign P3 fragment instead of amyloid beta. So gamma secretase cleavage of either membrane bound CTF also generates a cytosolic element, that is AICD, and APP also is an intracellular domain, sometimes referred to as CTF gamma, which will play a role in signal transduction. That is because of the essential role in generation of amyloid beta. Both beta and gamma secretases are considered to be prime targets for development of anti-AD pharmaceuticals. So hence preseralinin PS1, PS2, the enzyme complex and likely contains one copy of its subunit that is responsible for cleavage of multiple membrane proteins in addition to APP. And although the exact functional roles of its component have yet to be fully elucidated, but penicillin is believed to be a form of active site of aspartyl protease and nicastrin likely substrate docking subunit. Hence, explaining the gamma and beta, you do better understand with the video in the next slide. But the explaining the major form, I would let you know about the enzyme responsible for amyloid beta is production of BAC1 and BAC2, which is highly expressed in brain, but is also found at lower levels in other organs. So in contrast, to easily understand, the second form of the enzyme, BAC2, is low in brain, but is present in most peripheral tissues at higher levels. But the knockout of BAC1 in mice, in animal experiment in the mice, it led to a massive reduction in the level of downstream products of enzyme amyloid beta and CTF beta in brain. But in according to the human experiment, it's also the same. It highly increased, like level 0.1 to 0.2. But although these studies indicate that BAC1 is the major beta secretase activity in brain, some residual activity might be attributable to BACE2, and both forms of BACE can compete for substrate. So I'm playing a video. you can understand from this as a better the molecular characteristics, you'd better understand with the video.
Of Alzheimer's disease and its development and progression. And what we're going to find is that there are really two competing hypotheses, and most likely, just like in any case, it's probably a combination to some extent of both of these. Two hypotheses regarding how Alzheimer's develops and how it ultimately leads to deterioration of the brain, i.e. neurodegeneration. And one of the hypotheses, this is going to be the formation of amyloid plaques through the protein called beta-Amyloid, which is an abnormal protein. We'll discuss this second. The first one we'll talk about is the tau hypothesis. This is going to be the formation of neurofibrillary tangles due to the aberrant folding of the protein called tau. And there's going to be different reasons for the production and misfolding of both of these. But in any case, very similar to what we talked about in the introductory video to this playlist, we're going to have normal native structures of these proteins, and then by some mechanism, they're going to misfold into their prion form, which is going to propagate the disease of Alzheimer's. And really, in the normal sort of native healthy state of the protein, there's going to be more, relatively speaking, alpha helices in terms of their secondary structure. But when they misfold and you develop the prion form of the protein, you sacrifice some of those alpha helices for beta strands. So it's just a different conformation of the protein, but those beta strands are going to lead to the accumulation of that protein and therefore propagation of the disease. So again, the two hypotheses are the tau hypothesis and the beta-amyloid hypothesis. But keep in mind, those beta strands are going to promote the plaque formation that we're going to see. All right, the first hypothesis is the tau hypothesis, and this hypothesis states that Alzheimer's progression and development is due to the misfolding of a protein called tau. So let's talk about what tau normally does. So in the cell, we have structures called microtubules, and microtubules are very important for regulating things such as cell division and intracellular trafficking, things mainly associated with the nucleus, although there's other functions. And microtubules, whenever they form, they actually extend in length. So you'd actually extend the microtubule upwards like this, but they can also contract, in which case the microtubule essentially will fall apart. The process by which microtubules fall apart is what's called catastrophe. Okay, kind of seems like a harsh word, but when the microtubule shortens, it essentially just falls apart, and it's called the microtubule catastrophe. Okay, what tau normally will do in its phosphorylated state is it will stabilize the microtubule in whatever length it is and prevent a microtubule catastrophe. If you were to remove tau, the microtubules will fall apart. And so we have this protein called tau protein kinase 1. This actually is the same enzyme as glycogen synthase kinase 3. It just gets a different name in this context. And what it does is it phosphorylates tau to generate tau phosphate. Okay. And in the phosphorylated state, tau protein is able to stabilize these microtubules. But tau should not be hyperphosphorylated, meaning it should not be excessively phosphorylated. It has to have just about the right amount of phosphorylation. If it's not phosphorylated, it won't stabilize the microtubules. But also on the flip side, if you phosphorylate it too much, it will fail to stabilize the microtubules. And so this hyperphosphorylation of tau is what ultimately leads to, it's the initial stimulus that leads to the failure of these microtubules and therefore Alzheimer's disease. So what we do know is that inflammation of some kind leads to the hyperactivation of these kinases, the kinases that phosphorylate the tau protein. And so when the inflammation hyperactivates the kinases, that leads to hyperphosphorylation of tau, and we just call it hyperphosphorylated tau protein. In the hyperphosphorylated, meaning excessive phosphorylation, the tau protein fails to stabilize the microtubules, meaning the within the cell, which would include cell division and other functions such as trafficking within the cell, which is very important, obviously, for the normal physiological healthy function of the cell. So in the hyperphosphorylated state, these tau proteins not only fail to stabilize the microtubules, they also can oligomerize. And so we have tau oligomerization as shown right here. And eventually these oligomers will form sort of a plaque, as we would call it. Plaque is normally reserved for the beta-amyloid hypothesis as a term, but essentially you're going to form large aggregates. And in the context of tau, these have historically been called neurofibrillary tangles. And these tangles, which are very large aggregates of tau, can exist on the intracellular side of the cell, in which case they induce cell death, but they can also be exported out of the cell where they will themselves induce inflammation, mainly by their effect on microglial cells, which are just glial cells of the brain and the central nervous system. And whenever these neurofibrillary tangles of tau protein activate microglia, those microglia will release pro-inflammatory mediators such as interleukin-1 beta, which are known to even further propagate this inflammation and therefore hyperactivate these kinases, which lead to more hyperphosphorylated tau. So you can see here, this model proposes that this tau protein, in terms of its function in Alzheimer's disease, is going to be a positive feedback cycle. In addition, more inflammation can also trigger these kinases, and that's inflammation independent of these neurofibrillary tangles. And these neurofibrillary tangles, in inducing cell death, as I mentioned in this introductory video, will ultimately deteriorate the brain, particularly in the cerebral cortex area. That's the major part that's affected by Alzheimer's disease. You can see that with the deterioration of the brain, this neurodegeneration, the Alzheimer's brain is overall smaller than a normal healthy brain without Alzheimer's. And that's because of that neurodegeneration, which is induced by cell death. Again, the mechanism itself is not completely understood, but there is evidence that tau protein does play a role in this. This is the tau hypothesis for Alzheimer's disease. But as I mentioned, we also have a second hypothesis, and that's the beta-amyloid hypothesis. And here's a really good representation. I like this figure right here. We are not going to focus on the non-amyloidogenic pathway. This is the pathway that's the healthy pathway, which does not produce beta-amyloids. But we are going to focus over here on the amyloidogenic pathway, which just means the pathway that will produce amyloid plaques of beta amyloid or amyloid beta, and then this will lead to the progression of Alzheimer's disease. So there's a protein that normally exists in the plasma membrane of cells. It's called amyloid precursor protein. And amyloid precursor protein can be cleaved by several different enzymes. They're called secretases. And there's an alpha secretase, a beta secretase, and a gamma secretase. So if beta and alpha secretases, if these two forms of the enzyme are to cleave amyloid precursor protein, it tends to cleave in this way over here on the right, and ultimately you end up with this protein called P3, which is not known to be involved in Alzheimer's. This is the healthy pathway, you could say, that is non-Alzheimer's producing. This is what you want to happen. However, for some unknown reason, it's known that when both beta secretase, but now gamma secretase, when they cleave amyloid precursor protein, it cleaves it in a different way because alpha secretase and gamma secretase cleave at different locations. When beta and gamma secretase cleave APP or amyloid precursor protein, it cleaves in this way over to the left. And ultimately what happens is you get two major proteins that come off of this. You get this AICD, which is the amyloid intracellular domain. We don't really care about this. In fact, it really doesn't have any known function, at least in Alzheimer's, but the major one that we really care about is amyloid beta or beta amyloid, and it's typically abbreviated as A beta. This is the protein that will actually misfold. It will oligomerize and form what are called amyloid plaques. All right, so amyloid beta, as shown right here, can misfold. It's very prone to that. It will oligomerize and form alpha or A beta or amyloid beta oligomers, which can then aggregate and multimerize into what are called amyloid beta fibrils. You can see here, it calls it an oligomer, but they can be very large into fibrils. And these fibrils are going to be known to disrupt calcium homeostasis. induce neuronal destruction and ultimately apoptosis of that neuron. The mechanism by which they induce calcium disruption is not completely understood, but here's one model that shows how they may actually interact. So what we can have is a healthy protein. This is, notice the PRPC. This is the healthy form of the protein. It's actually going to be embedded in the membrane. However, these amyloid beta fibrils can actually interact with this protein. This protein has been thought to interact with a more transmembrane and intracellular protein called caveolin-1, which can then activate this protein called FYN, which can then phosphorylate and activate the NMDA receptor. Okay, this is one mechanism by which it has been proposed that amyloid beta can actually induce disruption of calcium homeostasis, considering the NMDA receptor, when glutamate binds to it, actually induces calcium influx into the cell. And what we hopefully have learned through previous videos is that whenever calcium influxes into the cell too much, you get cytotoxicity and ultimately mitochondrial dysfunction, which will actually lead to apoptosis due to the leakage of cytochrome C into the cytoplasm, which is a direct stimulator of apoptosis. What we also see here is that this FYN, this FIN protein, can actually phosphorylate the tau protein. So this is an additional kinase independent of the tau protein kinase 1 that can actually phosphorylate tau, leading to increased phosphorylation. And so you can see there are some crosstalks between these two hypotheses, or two pathways, if you want to call them that. Additionally, notice that amyloid beta is also an inflammatory stimulator of these microglial cells, which induce these microglial cells to release more interleukin-1 beta and induce more inflammation for more activation of these kinases, which lead to more tau phosphorylation. Now, my honest theory, given my ignorance on the topic, would be.
So we are next moving to in this video, we'll review about the amyloid beta generation from APP and APP and amyloid beta endocytosis, trafficking in the cell, and degradation with the lysosomes, amyloid beta induced cell death, and amyloid beta mediated neuroinflammation. So you could understand about the mechanism of Alzheimer's disease with amyloid beta plaques. So let's understand with this video.
Following synthesis in the endoplasmic reticulum, APP is transported to the trans-Golgi network. The newly synthesized APP protein is transported from the TGN to the plasma membrane via the endocytic pathway. APP undergoes clathrin-mediated endocytosis and is recycled back to the trans-Golgi network through the endocytic pathway. The retrograde transport of APP from the early endosome to the recycling endosome occurs through the interaction of APP with the retromer complex. Under disease conditions, APP at the plasma membrane is sequentially processed by beta and gamma secretases to generate amyloid beta fragments. This is referred to as the amyloidogenic processing of APP. Amyloid beta fragments have a propensity to aggregate and form oligomers, which eventually form large proteinaceous deposits known as amyloid plaques. APP, along with beta and gamma secretases, can be transported to late endosomes for processing. A subset of amyloid beta peptides are recycled back to the surface and released into the extracellular space. Another subset of these amyloid beta peptides is transported to the lysosomes for degradation. Amyloid beta and amyloid plaques bind to several receptors on microglia, which help clear pathogenic aggregates. Individuals with Alzheimer's disease express variants of the microglial receptor TREM2 that have reduced ability to bind to amyloid beta complexes. Amyloid beta can also drive persistent neuroinflammation by binding CD36 on microglia and rupturing lysosomes following internalization, which causes the production of inflammatory cytokines such as IL-1 beta. Amyloid beta oligomers interact with different surface receptors and can re-enter the cell via several mechanisms. Amyloid beta oligomers can mediate the formation of P75 NTR sortilin death receptors and also create pores in the plasma membrane. This results in ER stress or mitochondrial-mediated activation of caspases, which triggers the cleavage and aggregation of tau. It's the cleavage and aggregation of tau. Amyloid beta can also bind to low-density lipoprotein receptor-related protein 1, leading to endocytosis through the clathrin-dependent pathway. It can then traffic to the lysosome for degradation. Here, we reviewed how amyloid beta is generated from APP on the cell surface, and how these proteins are taken up, recycled, and/or degraded in the cell. We also covered how amyloid beta triggers caspase-mediated cell death and neuroinflammation.
Thank you everyone for looking for a video for a while. So let's move to the conclusion. Understanding the condition of amyloid beta is important for reasons other than intellectual ones, but clinical conceptions still strongly rely on classical pathology. But since they have not kept up with molecular knowledge, but given the difficulty in identifying clinically relevant biomarkers of Alzheimer's disease progression, with novel criteria such as specific molecular polymorphic forms of amyloid beta need to be examined. But the creation and application of imaging marker is one area where it is crucial to take various states or pools of amyloid beta into account. But the experiments with PIB and related benzothioline radioglands have shown that amyloid beta fibrils have several binding sites with various molecular specificities. And on fibrils, the site ligand stoichiometries vary and some of them seem to partially overlap. So the problem then becomes one of pharmacology, but it turns discovered that PIB binds with a high affinity to a site that is different from the site where Congo RAID and DDNP derivatives bind. But in the affected areas of Alzheimer's disease in the brain, this high affinity PIB site is present with high stoichiometry with respect to amyloid beta. But only very low stoichiometry PIB binding is seen in unaffected regions of Alzheimer's disease brain or any region of plaque-rich transgenic mouse brain or in non-human primate, temporal or parietal cortex containing similar amounts of insoluble amyloid beta. But the type of amyloid binding ligand used is important. Because of the plethora of new imaging molecules with different structures proposed for clinical imaging, studies may not be reporting the same form of amyloid beta, but much less as a disease-related one. But the uncritical use of such agents could potentially generate further confusion in the literature. But on the other hand, these ligands could be quite useful, providing that the binding site selectivity has been characterized under in vivo imaging-like conditions. Hence, my other papers in Alzheimer's disease is Alzheimer's disease neuropathological diagnosis, an extensive evaluation and meta-analysis of transcranial magnetic simulation, effectiveness as treatment of Alzheimer's disease, and lapse memory deficiency in Alzheimer's disease, model using physical and cognitive exercises, You can find all these presentations in OLCAS website in YouTube. Interested people can find these papers in ResearchGate and open request on my email ID. And thank you very much. This is from my side. Thank you.
All right. So this was the presentation by Dr. Tshetiz Dahal, which was on beta-amyloid peptide and Alzheimer's disease. Thank you so much, Dr. Dahal. And I would like to share that our Dr. Dahal is also one of our OCMs. Yeah, Dr. Dahal, there were two questions from Dr. Ranjana Dahal earlier. If you would like to take the questions, please. And if you want me to repeat the questions for you, I will. I hope I'm audible to you. Hello?
Yeah, you're audible to me.
Yeah, thank you, doctor. There were two questions from
Okay, sure.
Dr. Ranjana Dahal.
Okay.
Welcome to the event, Dr. Dahal, Dr. Ranjana Dahal.
Ghazal, actually, she is my sister. She's listening you from Haryana, India.
Oh, that's great.
I invited her to our event. Actually, okay, Ranjana Dahal, the risk of developing Alzheimer is somewhat higher if a first degree relative, your parent or sibling, has the disease. But just how genes among families affect the risk is largely unexplained. And the genetic factors are likely complex. But a better understood, a genetic factor is a form of apoprotein E, that is APOP gene. So like parent is like people who develop Alzheimer do not always have a history of disease or their families, but still those who have a parent or sibling diagnosed with the disease have a higher risk of developing Alzheimer's than those without that association. Hope you understand. So the next question is, what is the relationship between aluminium and the development of Alzheimer disease? Okay. So relating with the aluminium. Okay. So aluminium, mainly it's the level concentration in people brain that hasn't been shown to be toxic, but an age, okay? But the aging of a brain that may be less able to the process the aluminium, although the aluminium has been seen in amyloid plaques, but there is no social evidence that aluminium is increased in brain of people with Alzheimer disease. But your question is how aluminium affect, and those are the physical properties. But aluminium and ferric ions are similar, allowing aluminium to use mechanisms evolved for iron to enter vulnerable neurons that may be involved in Alzheimer disease progression, but they may accumulate in those neurons and cause neurofibrillary damage. That's all. Thank you.
Yeah, so thanking Dr. Dahal, but I would like to thank Dr. Ranjana Dahal first. In my views, it came up like Dr. Ranjana Dahal is asking the questions for us from our behalf to Dr. Dahal, and thank you so much for that. Yes, Dr. Sampurnam, it was indeed very informative. And yeah, please call me Ghazal. It's Ghazal Johar.
Thank you, Dr. Sampurnam. I would appreciate from a person like you.
And there's one more question for us from our Dr. Ranjana Dahal. The question is meant for us.
Okay, what is
And
the difference between
yeah.
normal memory loss due to aging and memory loss associated with Alzheimer's disease? Okay, your memory often changes as you grow older. So, but memory loss that disrupts daily life is not a typical part of aging, but it may be a symptom of dementia. So, the dementia is a slow decline in memory, thinking, and reasoning skills. That's all. Thank you.
Yeah, thank you for the question, Dr. Ranjana Dahal, and thank you, Dr. Dahal, for answering the questions. Let me ask any other questions from our other attendees and speakers. If there is, please come up, raise your hands and ask your questions. We are glad to have you, Dr. Ranjana. So I think it was Ms. Sakshi who raised hand earlier. So was there any question from your side, Ms. Sakshi? There's one more question.
Okay, so what other conditions can mimic Alzheimer disease? Okay, so the other conditions are like thyroid, kidney, liver, heart, and lung problems. Urinary and chest infections and strokes are among the many medical conditions that can produce dementia-like symptoms. But mainly people mostly common misdiagnosed with FTD, that is frontotemporal dementia, that is misdiagnosed with Alzheimer's disease, like psychiatric disorders, vascular dementia, or Parkinson disease. So I've already explained the early symptoms. So that's all. Thank you.
Yeah. Dr. Dahal, I have a question that is a very basic one.
Okay.
As we had a conversation earlier, and you said that you're a big fan of yoga, right?
Yeah.
Yeah. So, Dr. Dahal, is there any particular yoga move that is effective for such conditions that you?
Yes, sure. Because, like Alzheimer disease, you can practice like yoga, meditation, and pranayama. That can
Yeah.
bring a much needed and valuable gift to those suffering from Alzheimer disease. Those practices can not only often like stress relief, but lead to a reduced cognitive decline, improved memory, and diminished depression, as well as soothe hyperarousal. That is
Yeah.
because of yoga.
So, any particular move, any particular yoga move you would suggest?
No, actually, I don't know about the asanas for Alzheimer disease. Maybe you could consult with Baba Ramdev.
Oh, he is very far in reach. I mean,
Yeah, but according to my research, yoga and meditation can play a prevention and improve symptom, but quality of life of patients are caregivers. So just
yeah.
practice like, pranayama and meditation, but there are no such asanas yet developed, or I don't know about the yoga, but still, when I know about it, I'll let you know. Sure.
Okay. Thank you so much. I'll take it answer as a pranayam and meditation. Yeah.
Yeah.
Thank you. I'm gonna try it and definitely
Yep.
let you know what changes I saw in my behavior in my day-to-day life. Yeah.
Okay, sure. Thank you.
Thank you. Thank you, Dr. Dahal. So with your permission, let's move forward.
Yeah, sure.
Yeah. Thank you. So everyone, let's move to our third speaker of the day. It's Dr. Amara Rafiq, Department of Biochemistry, University of Karachi, Pakistan. And the title of her talk is cognitive rehabilitation of cerebral palsy patients using negative air ions. Dr. Amara Rafi.
Yes, can you hear me? Hello.
Yeah, it's clear.
Okay, and my screen is visible to each and everyone?
Not yet. I'm sorry, doctor. And welcome to the event.
Thank you. Okay, I'm sharing my screen.
Sure.
Is it visible now?
Give me a while. Okay, doctor. It's visible now. Can you please maximize the slide?
Yes, I am doing it. Yes.
Perfect.
Okay,
All the very best to Dr. Amara.
thank you. Hello, everyone. I'm Amar Rafiq. I'm a PhD researcher. I have done my PhD in biochemistry from University of Karachi. And I'm also serving as a teaching associate at the Department of Biochemistry, University of Karachi. And I'm also visiting faculty in Indus University of Health Sciences, where I'm teaching biochemistry to physiotherapists. So the title for my talk today is Cognitive Rehabilitation of Cerebral Palsy Patients Using Negative Air Ions. And the cognitive rehabilitation is basically a term which is used to define a therapy which is aimed to improve or restore cognitive functions or thinking skills after any brain injury or illness. Basically, in cerebral palsy, there is a brain injury which causes a deformation and which leads to physical and mentally deficits. So it is actually cognitive rehabilitation is actually helping in treating various conditions that affect cognitive functions, such as traumatic brain injury, stroke, brain tumor, epilepsy, and even in cerebral palsy. So it involves practicing skills, learning techniques, or establishing new or compensatory patterns of cognitive activities. So moving to the term cerebral palsy, maybe it's a new term for many of us. So I would like to introduce the term first, that cerebral palsy is basically a non-progressive neurological disorder which is caused due to formation of abnormal tissue in the brain before birth, during birth, or shortly after birth. There are several risk factors for that. I'll discuss those as well. And worldwide, it affects 1.5:3 per thousand live births. And unfortunately, in Pakistan, we don't have any established CP morbidity data yet. And even in India as well, to the best of my knowledge, there is no such data reported yet. So we need to work on that particular area. The CP characteristics involve basically that it affects the movement, feeding, posture, communication, behavior, and it can induce a seizures, that is basically fits, and it affects cognition in almost 50% of cases. So the cognition is the main focus that I am going to discuss in this particular study. CP is classified on the basis of topography, the muscle tone, severity, and the gross motor function classification systems from level 1 to level 5. There are several reported risk factors that can cause cerebral palsy, and those risk factors involve head injuries, multiple or twin babies, brain infections, low birth weight, genetic predispositions, and preterm birth, intracranial hemorrhage, or bleeding into the brain. There can be maternal factors, including maternal diabetes, hypertension, or malnutrition during pregnancy. And severe jaundice can be physiological or pathological, or due to head injuries, like due to sudden TBI, that is traumatic brain injury, sudden fall, sudden accident, or due to child abuse as well. Now, I would like to discuss the term negative air ions. It might be new for many of us. We know that negative air ions are beneficial, but when it comes to rehabilitate the patients, particularly CP patients. This is the research that was not done before my research. Back in 2021, when I started this rehabilitation study, I searched thoroughly and I found out that no cognitive rehabilitation study has been performed yet for CP patients. So I initiated this study, and I saw that several studies have performed and reported beneficial effects of negative air ions in increasing productivity, physiological health, and well-being. And those studies use the concentration of about 1600 to 1500,000 ion concentration per centimeter cube for different for different intervals. And we basically used a negative air ion generator. It's a device which can produce negative air ions, and these negative air ions are also abundant in our nature, and likely around sea, green mountains, waterfalls, rain, or forest. So there are also several commercially available products. Maybe most of you uh, don't know that there are Himalayan salt lamps, there are Himalayan salt, rock salt as well, which can produce negative air ions. So there are also energy stones and special type of bracelets that can produce negative air ions. We often use negative air ionizers in our cars. They are portable devices. So there are several proven effects of negative air ions on humans. I have used this source, and it has a beneficial effect on nervous system, fatigue, blood pressure, pulse, oxygen consumption, balance, metabolism, pain, allergies, nasal congestion, and the journal capacity of person. The Limosity software is a software which I have used for the evaluation of cognition in the CP patients. That's why I would like to discuss what is Limosity software. It might be a new term for many of us. So the Limosity software is basically a software which is developed by Lumos Labs in San Francisco. And scientists have conducted a randomized trial with almost 4,715 patients, and they have reported improved cognitive performance after training with this software. So studies have also indicated that this training, the cognitive training through Limosity software, actually improved the cognitive function of healthy adults and early stage Alzheimer's patients. It's a very great study. I have also mentioned the reference for this study at the end. And so based on that idea, I thought of employing this Limosity software for the cognitive training of CP patients. The purpose of this study that I am going to discuss in this was that to determine the effects of negative air ions on the cognitive function of CP-inflicted patients. Our methodology was that after taking the ethical approval from IBC, that is our university's ethical bureau, we took consents from the participants, and 30 parents gave us consent for inclusion of their children in the study, but 2 irregular patients were excluded from the study. We used a concealed method of randomization to allocate 28 patients in 2 groups, and in control group, we didn't give any intervention, but in intervention group, we gave an exposure to negative air ions along with their regular therapies. And there was a pre-evaluation of 5 Limosity games, that is at baseline, before dividing them into 2 groups. And then after the provision of intervention, we again did post-evaluation of the patients, and 5 Limosity games were again played by them. So the statistical analysis were also performed to see the difference. The selected games were selected by the professionals that are dealing with the CP patients at Alumni Rehabilitation Association, where the study was conducted. I would like to tell that Alumni Rehabilitation Association is the biggest institute, which is a rehabilitation center, basically, which is dealing with CP patients solely CP patients in Pakistan. So we took permission from them and we carried out our experiment there. So their professionals selected 5 games for the cognitive evaluation, which included highway hazards, lost in migration, masterpiece, space trace, and tidal treasure, which allowed us to, assess the information processing skills, selective attention skills, spatial reasoning, spatial fluency, and working memory in those patients. These all skills are basically help in the cognitive in the evaluation of cognitive skills or cognitive functions. Now the exclusion criteria were that we only included those patients that can hold the computer mouse using their hand or feet. You can see here a patient who had so much spasticity in his arms, so he was using his feet to control mouse and the keyboard. We also include the inclusion criteria was also that the patients who can actually understand the instructions that are given by the researcher. And the exclusion criteria were that the patients who have photosensitive epilepsy. So we excluded them because we didn't want any problem for the patients. Now the intervention study was that we actually bought these negative air ionizers and we used that in the room. And I have mentioned here, I have encircled the negative ionizer. We actually placed 4 patients in a room at a time, and the room was enclosed to have a constant amount of negative air ions in the room during the whole session. We actually started this process and in their rooms and without any air condition or any other problem, so that the concentration of negative ionizers is constant during the process. It was a 40-minute session of negative air ion exposure to the patients, and into that session there was no problems of air passage or air condition or anything, and the concentration of negative air ions were detected using this device that was KT401, and we actually calculated we actually monitored the concentration of negative air ions at the start of the session and at the end of the session, and it was kept constant at 10,000 ions per centimeter cube, and the room was of dimension were of 7 feet by 7 feet. Now, for the statistical analysis, pair T tests were performed. Now, the results that we observed that at the 6th week, the control group had only improvement in the spatial reasoning, but in the intervention group, which was provided that negative air ion intervention for 6 weeks, they had a great improvement in their cognitive functions at the 6th week. And the selective attention, information processing, spatial fluency, and spatial reasoning skills were significantly improved at 6th week. Now the discussion. Serotonin is a neurotransmitter which plays a vital role in regulating endocrinal, metabolic, neurovascular, and behaviors that have higher cognitive demand. So the serotonin neurons in the frontal cortex regulate cognition and memory. So we envisage that inhaled negative ions can stimulate biochemical reactions upon entering bloodstream, which increase the 5-HT levels in the brain. There are actually the studies which have mentioned that negative air ions can actually stimulate biochemical reactions upon entering bloodstream. So this might be the reason for improvement in our patients as well. So these increased 5-HT levels can raise the flow of oxygen in the brain cells, which improve cognitive alertness, mental clarity, and information processing ability. So we think that the improvement in cognitive functions may be due to enhanced neural processing or alertness status in an environment in which it NIIs. So there may be possible stimulation of frontal cortex, parietal lobe, temporal lobe as well. which improved the cognitive functions in these CP patients. Of course, we didn't monitor the serotonin levels in these patients, but it can also be done in future prospect. So based on our findings, we concluded that exposure to negative ions can improve the cognitive functions in CP patients. But the limitations were that due to less amount of patient, it was a first clinical trial, and second and third phase clinical trials can also be inducted with a bigger massive scale of patients to further confirm our reported findings. And we recommend that a longer duration of intervention may also improve working memory, because in this particular case, there were improvement in 4 of the cognitive skills, but not working memory. So maybe a longer duration of intervention can improve that as well. Our intervention was only for 6 weeks, but if we can, use a longer duration, maybe the working memory can also be improved. So based on our results, we also suggest that this intervention can be a miraculous, complementary, non-pharmacological, economical, and an effective approach for the cognitive function betterment of the CP patients. I would like to acknowledge Alumni Rehabilitation Association, which allowed us to perform study at their center. And these are the references which I have used in the particular study. Thank you, everyone. That was a very short talk.
But it was quite informative, Dr. Amaro. Thank you so much for explaining the cognitive rehabilitation of cerebral palsy using negative air ions.
Yes.
It was quite informative. And to compliment you, I would call Dr. Tshetiz Dahal, as it is, as far as I know, if I'm not wrong, it is Dr. Tshetiz Dahal's favorite topic. And he has already chaired an event, and you were a part of it, if I'm not wrong.
Yes, I did give talk on that particular topic as well, on CP patients. Yes, I was there.
Exactly. Yeah, exactly.
Yes.
Yeah. So it was, and there was Dr. Zahan Juan Liu also, and Dr. Tshetiz Dahal has chaired the whole event.
Yes.
It was, yeah. Let me ask Dr. Dahal to compliment you. And if he would like to add on something.
Yes, why not?
Yeah. Thank you. And hello, Dr. Dahal.
Sir is asking that what is cerebral palsy cognitive? Cognition is basically disturbed in CP patients. So that's why for that rehabilitation of cognition, we have employed this particular intervention. So yes, studies have shown that cognitive deficits are present in almost 50% of CP patients. So it is a very big, there was a big research gap in that area, that something needs to be done, something needs to be, to have intervention to have rehabilitation of these patients. So I assessed cognition in CP patients using that Limosty software games. We have actually done pre and post evaluation of the cognitive scores in those 5 games, and seeing that there was a difference, big improvement in the patients that were given intervention. I have discussed that in the result section, that after 6 weeks of providing the intervention to those patients, there was a cognitive improvement in those patients. And the assessment was done using that Limosty software and those 5 games. Is my answer clear to you, sir?
Yeah, it's clear. Thank you.
Welcome.
Dr.
So,
Dahal, I'm sorry for the interruption.
okay,
Yeah.
so can you explain me, like, what are the 4 basic cognitive concepts in CP?
The basic concept, sir?
Yeah, the 4 basic cognitive concepts, not the concepts, but the cognitive concepts.
Yes, we have done that. That in the studies have explained that in the patients of CP, there are disturbances in the cognition, and in cognitive functions, basically, we have only focused on information processing skills, selective attention, spatial reasoning, spatial fluency, and the working memory. So these games are particularly, which I have discussed in the methodology section, that these games are particular. Each of these games is actually assessing a specific cognitive function. That,
Okay,
like
I mean, like not
yeah,
the games, but the
yes,
cognitive concepts. Like I mean about like sensory motor intelligence. Have you heard about it?
yes,
Like
of course,
yeah, the pre-operational thinking
yes,
and concrete
yes,
operational thinking and format
yes.
operational thinking. Yeah, you should more work on that topic. That would be
Yes,
more
of course,
interesting.
I am. Yes, I am also designing a new research project for my postdoc. Let's see if I can work on it, because I am really looking forward to, assess if there is any assessment of neurochemicals or any other processes which we can see actual
Yeah.
what is happening in the brain.
Okay, I'm
So
working on a best drug that would improve memory in CP. That is
very good.
cholinesterase inhibitors. I hope you have heard about
Okay.
it. Yeah, I'm working
No,
on.
I don't. Okay,
Yeah.
very good. Very good. Interesting.
Okay, thank you. Thank you for the answers. Thank you.
Thank you so much, sir.
We are having another question also from Dr. Ranjana Dahal.
How does failure to properly interpret and act upon fetal heart rate monitoring cause cerebral palsy? Maybe I'm not getting the answer, the question, but I think she is asking about the maternal factors that are causing CP in the patients before birth, during birth, or shortly after birth. Maybe she is asking about that. Dr. Ranjana, kindly, can you please explain your question?
I think she is asking about the development of a baby brain while it's growing in the womb. That is a damage to the part of brain that is called white matter, possibly as a result of reduced blood or oxygen supply. That is also known as periventricular leukomalacia, PVL.
Yes.
Hello. Hope I'm audible to Dr. Amara. Hello, Dr. Amara.
Yes, I'm there.
I'm sorry. I thought it's again a network issue.
Yes, there was.
Yeah, that's why suddenly I started using the screen also. So thank you so much, Dr. Amara. Let me ask if anyone else is having any question from you. So dear attendees and other speakers, if you are having any questions to be asked from Dr. Amara Rafiq, please go ahead, raise your hand and ask your question. And also, thank you so much, Dr. Dahal, for both of our Dr. Dahals for the questions. And thank you so much, Dr. Amara, for answering the questions, taking the questions. Yeah. Thank you for the informative talk, Dr. Amara. So with your permission, let me ask our next speaker, who will be. Let me share the details also. So everyone, our fourth talk will be from Mr. Ayodeji Ayomikun, University of Ibadan, Nigeria. And title of his talk is Overview on the Role of Physiotherapy in the Management of Dementia.
Good evening, everyone. Can you hear me?
Yes, clearly. Good evening.
Good evening.
Yeah, welcome to the event, Ms. Ayodeji.
Thank you.
And so, would you like to share the slides?
Yes, I will do that. Let me go ahead to share the slides.
Sure. Till then, it would be great if you could share something about you, your short introduction from your side.
Okay, I'm a musculoskeletal physiotherapist in Nigeria. I am adjunct lecturer at the Department of Physiotherapy, University of Ibadan. Thank you.
Thank you. Thank you, Ms. Ayodeji. Yeah, please take your time.
Okay, I'm trying to share my screen, but it's telling me the documents can be shared. So I think I should go ahead.
You can, we have an option here. You can simply mail the slides to us and we will play the slides for you from our
okay.
side. Yeah.
Okay, should I send that to you now?
Yeah, let me share our official email address here and you just have to share your slides here and we will play the slides for you.
All right, thank you.
You're welcome. Here, I have shared the email address in the chat box.
Yes, I'll do that now. Sorry for the delay.
That's okay.
Sorry.
Please take your time. Be comfortable. I am sure everyone in the meeting room is well aware about the technical glitches, and it's okay. It happens with everyone.
All right, send. I think you should check
Okay.
the mail now. Yeah.
Okay, let me confirm with my team. Just a second, please. Okay, slides are well received and we will play the slides in a while.
All right, thank you. So I will be speaking on the overview on the role of physiotherapy in the management of dementia. Next slide, please. Okay, so why dementia? Dementia has been considered as the most severe type of cognitive impairment. And according to the WHO, it's the 7th leading cause of death. And then in looking at the global burden of dementia, the prevalence talks about that more than 55 million people have dementia worldwide. And then over 60% live in the low and middle income countries. And then in looking at the incidence presently, there are nearly 10 million new cases reported every year, according to the WHO. So what is dementia? I would say dementia is a syndrome of decline in memory and cognition. as a result of a brain disease. It means that there is something ongoing in the brain, and this results in significant impairments that affect important aspects of an individual. And that's the physical, the personal, and social, as well as the occupational function of the patients. The next slide, please. Thank you. So what are the risk factors? Age is a major risk factor for dementia, but it does not mean that everyone at the older age have dementia. It simply means it's more of a predisposing factor. And so family history as well predisposes an individual to dementia. And also gender and the sex. It's been reported that dementia occurs more in the females than the male. And also cognitive engagement affects an individual to having dementia. And so in talking about cognitive engagement, we are talking about when an individual leaves education early. Hello? Can you hear me?
Yes, Miss Ayadeji. I'm sorry for the disturbance.
Oh, okay. And when there is less job complexity of an individual, it could predispose the individual to dementia. And also ethnicity. It's been identified that people from the Black African, the Black Caribbean, and the South Asians are more likely to have dementia. And health conditions as well, such as hypertension, cardiovascular diseases, talk about stroke, talk about Down syndrome, obesity, and diabetes as well predisposes an individual to dementia. And also the lifestyle factors. You're talking about the individual who smokes, drinks, and sedentary lifestyle also predisposes an individual to dementia. The next slide, please. And so what are the stages? Because dementia has a gradual onset, but there is a progressive decline in memory, in cognition, and in other aspects that affect the individual. And so it comes in stages, and it's usually occurs over a period of time. And then under the advanced dementia, you could see that I highlighted the fact that it's usually about 10 to 12 years from the onset of symptoms. So it could take a while for an individual to reach the last stage of dementia. The next slide, please. So what are the clinical features? Well, I didn't mention the types of dementia because I think it's been mentioned mostly in the conference. And what I would like to point out to the fact that the types of dementia just simply talks about different pathophysiology and then clinical features that are peculiar to each type, such that you can have a particular clinical feature being seen more in a type of dementia than the other. And so what are the clinical features? The patient would experience pain, the patient will experience mental or cognition decline, and also the patient would experience depression, low motivation, and then frailty. Yes, frailty can be defined as a state of decline in multiple organ systems that is characterized by a state of low physiological reserve, loss of function, and an increased vulnerability in older adults. And also the patient would experience impaired balance. And when there is an impairment in balance, it affects the gait and affects the function of the individual. And so that predisposes the patient to fall a whole lot more, and also loss of function. The next slide, please. And so what are my goals as a physiotherapist? I am ensuring that my management is patient-centered. And so I am developing an individualized care plan for my patients. And my focus is actually to maintain or restore function and mobility in an individual in order to improve their quality of life. So usually, it's usually patient-centered. It means that the treatment protocol I designed for a particular individual who has a dementia might be a bit different from the other. So I'm looking at my patients as an individual and what is peculiar to them. The next slide, please. And so what does my assessment entail as a physiotherapist? My assessment is holistic. Earlier, I would, in defining what dementia is, I explained the fact that it affects certain aspect of an individual. And so in my assessment, I'm being holistical in the sense that I'm not looking at only the impairment of this patient. I'm looking at participation restrictions. I'm looking at the social component. I'm looking at the family history. I'm looking at the patient as a whole. And then in doing that, I am being multidisciplinary in my approach. It means that I am not the only healthcare professional that will deal with the individual with dementia. And so I work in collaboration with other healthcare professionals, and that's important. And so in assessing the patient with dementia, it's important to know the level of cognition of this patient. And this is because usually a cognitive assessment will indicate the level of mental capacity and the degree of memory loss for this patient. Because in seeing any other patient, not even a patient with dementia, it's important my patient consents to my management and interventions. And so usually in patients with dementia, they could have difficulty with decision making. And so cognition assessment, it's important in patients with dementia. And because this will influence my treatment protocol that is formulated for my patients. And so as well, in my assessment, communication strategies, they're important because I'm dealing with patients who might have difficulty in communication, difficulty in comprehending my instructions, comprehending my conversation with them. And so I have to look at ways how to communicate with my patients. In times where they don't understand, I will look at, how will I go ahead to ensure that I communicate this patient to communicate to this patient in a simpler way for them to be able to understand. And I'm also putting into consideration their family. Will there be any other person I could talk to that could tell me things I need to know about the patient? So in my assessment, communication strategies are key. And usually my assessment also entails a subjective and an objective assessment. A subjective assessment will entail the onset of the symptoms, the duration, is going to entail the medication, is going to entail the program condition, is going to entail a whole lot of things. I'm looking at the family social history. I'm looking at the social economic status of this person. I'm looking at this person as a whole. I'm looking at, what kind of environment do they live with? The accommodation, do they live alone? Do they live with the family? I'm putting that all in consideration. And also on that objective assessment, I'm looking at laboratory testing to rule out, does this patient really have dementia? Or does this patient have something entirely different from dementia? And so I'm doing my neuromuscular assessment, I'm doing sensory assessment, I'm doing all of that in order to be fit to effectively develop a care plan for my patients. The next slide, please. And so I identified some major clinical features, and then outcome measures, objective outcome measures that can be used in the assessment. And so here I have pain, firstly. And so there is an outcome measure called numerical pain rating scale, but this is usually used when the patient can comprehend my information. But in situations whereby the patient can't really tell me the severity of their pain, I would consider the use of a pain assessment in advanced dementia. So there are outcome measures that are being used in assessing the clinical, the rate, the level of severity in patients with dementia. And so for a mental decline, there are outcome measures listed there. There's a mini mental state examination. There's an Havenbrook's cognitive examination. There's a mini cog assessment instrument. And under low motivation, which could be as a result of depression, I could use the geriatric depression scale. And also frailty, like I spoke earlier, I can use the clinical frailty scale. Usually it's believed that the level of frailty in an individual usually correspond with the level of dementia. So it means that if my patient has a mild dementia, usually in the level of severity of frailty, they will also have mild frailty. And so I'm also assessing balance, because this is key for my patient, because an impaired balance will affect gait and then will affect and predispose the patient to fall. And so I'm also doing a 4-week assessment. 4-week assessment is a lot of components. I'm putting a lot of things in consideration to ensure that my patient isn't predisposed to fall. And so I'm also looking at the loss of function. I'm looking at the basic activities of daily living and also the instrumental. So I mustn't limit myself to the basic activities of daily living for these patients. Under the basic, I'm looking at, can this patient walk independently? Can they feed? Can they dress? Can they groom themselves? I'm looking at toileting, bathing, and transferring for these patients. Under instrumental, I'm looking at, can this patient manage finances? Can they manage communication? Can they use telephones to communicate? Can they do house cleaning, house maintenance, and all of that? So there are usually outcome measures to be used in assessment of patients. The next slide, please. And so what are my rehabilitation interventions for this patient? Depending on the clinical features and presenting complaints of my patients. Most importantly, I must educate my patients, and I must educate the caregivers, because they must be aware of what dementia entails, what they should expect, the prognosis, all of that. I have to ensure that my patients and their caregivers are aware. And we also have falls prevention strategies. Under falls prevention strategies, we are looking at strengthening exercises. Why? Because when there's an impairment in the muscle strength, it could affect balance, it could predispose the patient to fall. So I'm targeting strengthening exercises for my patients. Also balance training, gait retraining, and the use of mobility devices in situations where my patient can't stand or work independently. I would prescribe the right mobility device for the patient. And also I'm looking at environmental modifications, and this is usually done in collaboration with occupational therapists. Like I said earlier, the management of dementia is usually multidisciplinary. And so under environmental modifications, I'm also considering referral to the occupational therapist. And also electrical stimulation, because at the advanced stage of dementia, patients usually might have urinary incontinence. And so I could use electrical stimulation for pelvic floor strengthening exercises to improve my patients. And also massage therapy can be used to relax my patients from stress, from anxiety, and also aerobic exercises. I could prescribe patients to take brisk walking, the use of bicycle ergometer, just to lighten up their mood. And I could incorporate group exercises such that they are brought into groups and they're managed together in order to encourage each other, kind of. Thank you. The next slide, please. And so in conclusion, dementia has a health and social impact for patients and as well for their family caregivers, and this will affect their quality of life. And so physiotherapists, alongside other healthcare professionals, they help to improve the quality of life of patients. The next slide. These are my references. Thank you. These are my references. And the next slide. Thank you.
All right. So thank you so much to Ms. Ayadeji for sharing how the dementia can be managed by physiotherapy. I hope I'm audible.
Yes, you're audible.
Okay, so the voice changed. I mean, I'm sorry.
Okay, I'm here.
Yeah, I'm sorry. There was a voice change and I got confused that whom I was talking to at first place. All right. Thank you to the attendee who just confirmed that I was audible. All right. So, Ms. Ayadeji, there are some questions for you in the chat box. Yeah.
Sis, can you read them out to me?
Yeah, sure. So the question is from Dr. Tshetiz. The first question is, what are coping techniques for dementia?
Okay, coping techniques for dementia involves mechanisms that helps the patient to deal with issues arising from dementia. And then I would say majorly it's the coping mechanisms involve ensuring and encouraging the patient to keep an active social life that would help the patient. And like I earlier mentioned, regular exercise, I mentioned aerobic exercise can be a form of coping mechanisms for the patients with dementia. And so you also encourage the person to continue the activities they seem to enjoy. They seem to enjoy before activities they enjoy doing, and that would also help. And as well, you could encourage them to find new activities that could lighten up their moods and in a way would help reassurance for patients with dementia.
Okay, Dr. Dahal is also asking for 5 main coping strategies.
Okay, I think I've mentioned them. You would exercise, encourage the person to keep an active social life, a regular exercise, and then you encourage the individual to continue activities they enjoy and probably find new hobbies and new ones that could help their mood or help their cognition. So you're ensuring that they're consistent with their social life, keep up with exercise, good nutrition, good diets. They are all part of the coping mechanisms for patients with dementia. And also in cases where you identify that there's a particular health condition that has predisposed this patient to dementia, you would encourage that probably alcohol consumption, you encourage they limit it and they quit smoking. And then in situations where they have high blood pressure, you are also encouraging them to control their blood pressure. I hope I've done justice to that.
Let me ask Dr. Dahal. Was it satisfying, Dr. Dahal? I hope I'm audible to Dr. Tshetiz Dahal. And there was one more question. I mean, the third question from Dr. Dahal. It was 2 particular treatments that may help with cognitive rehabilitation.
I think it will have to, 2 treatments will be as regards keeping them around their families, ensuring that they are not isolated. They are not isolated from the social environment. And so I think that would help them. You also get them involved in activities that could also help their cognition. Maybe if you could read out to them, you could help them just to restore some lost function they could have. And it's usually goal-oriented, depending on the patient need. I think that's what I would say. So you are educating the patient, you are considering all of that to ensure that there's a restoration in cognition.
Yeah, thank you so much
And
for taking
like
the
I
question.
said, I think the occupational therapy. Yeah.
All right, thank you so much for taking the questions, Ms. Ayodeji. All right, Mr. Shahid, I'm sorry, but the questions have been already discussed in the very first introduction from Dr. Sampurnam Webster. And please don't worry, we will definitely share the recorded introduction also on the YouTube, and you will be able to watch that introduction later on our YouTube channel or in the discussion portal of this very particular event. Yes? Will that be okay, please? Okay, thank you. So, any more questions from Ms. Ayadeji? If you are having any other questions to be asked, please go ahead. All right. So while thanking Ms. Ayadeji Ayomi-kun for her talk, let us call our next speaker. It's Dr. Abolaji Erinfolame.
Hello, can you hear me? Hi.
Hello. Hello, doctor.
Hello.
How are you?
I'm fine. Good evening, everyone. How are you?
I'm good too. Thank you for
All right,
asking.
so go ahead to share my screen now.
Yeah, sure.
Okay.
So everyone, here is our Dr. Abolaji Erinfolame from Neurodiagnostic Elite Healthcare Professional. He's from Nigeria. And title of his talk is Electroencephalogram, Nerve Conduction Study and Memory Disorders.
All right, thank you, Gazal, for the introduction. Hi, everybody. My name is Abolaji Erinfolame. I'm from Nigeria. I'm a neurodiagnostic and respiratory therapy allied healthcare professional. So basically, I'll just try to talk about the unraveling mysteries of memory disorder, diagnostic and research approaches. I'll be focusing my talk basically on an electroencephalogram, that's the EEG, the nerve conduction study, and how it relates to memory disorders. All right, so basically we'll just start out with a little bit of a view of what I know most of us know already, what memory disorder is all about. A neuroanatomical structure that in that storage, retention, and recollection of memories. We know it can be progressive, like Alzheimer's disease, and that can have immediate. We also know we can have the ones that have immediate effects or disorders resulting from head injury. We also know to a certain degree that they can be defined as suppressed cognition abilities leading to deteriorated ability to reason and make decisions. Can also lead to communication hindrance, also making the person or the one that has the complication not being able to remember stuff correctly or speak out correctly. That's basically all the things that complications that surround memory disorder as a disease. So I know we all know a lot about that already. I'm going to go to, basically what I want to base my talk on. So we'll start with what we call the electroencephalogram, also called the EEG for short. That's advanced neurological procedure, which records electrical activity of the brain. It's a non-invasive procedure, which means it's not a painful procedure. We don't have to have direct go into the beyond the scalp of the patient. So it's not painful in any way, actually. So basically, we have it for those that have detected of seizure and also epilepsy. Is one of the major tests to do for them to kind of clarify and ascertain where the seizure or epilepsy is probably being triggered from. Of course, we have a lot of functions that the EEG can also, carry out, can help us to diagnose epilepsy or memory disorder or memory loss as a result of head injury, maybe dementia, brain tumor, infection, stroke, or sleep disorders also. So moving forward, now what we have here is like a picture representation of what the EEG looks like. We assume that the circle we can see here is actually the patient's scalp. I usually advise that if a patient is coming for the EEG procedure, he or she should have their hair cut down so we can actually have access to the scalp, where you can see here where we're placing the electrodes, right? You can also see, I tried to put down here what the abbreviations mean. We have the FP, which is a frontal polar. We have the F, which stands for frontal. We have the C, which stands for central. We have the T, that stands for temporal. We have the P, that's parietal. And of course, the A, that is referenced. Now, basically, all these are just points on the scalp where we place the EEG electrodes before we begin the procedure. And of course, we have a placement system universally acceptable, which is the 1020 International Electrode System of Placement. And you can see here that on the left side, you can see that we have some electrodes being shown here on how it is being placed. And we can also see here on the right hand side that we have a different kind of view of electrodes that are being placed. Now, this is more or less like this is just to accommodate differences that EEG machines usually have. Some of them have a lot of electrodes that might look like this, and some have less electrodes that actually look like this. So it's like one doesn't get confused on how to apply them. This is actually what it looks like. So in a situation where we have abbreviations like the FT would tell us that we're talking about frontal temporal. Like here, where we just have them as one, we have F, we have T, we have Z, we have P, and we have O. They all separate, standing separately. But you can see here that there's a little bit of, there's more electrodes here. We have more electrodes here. We have the CP, which is actually not present here. So that tells us that we have some EEG machines that can actually, that kind of have more electrodes, and this diagram here kind of accommodates that, kind of tells you what it means, right? We could have electrodes that could be central parietal, unlike here where we just have it, we don't have that. We have just central and we have parietal, but we can see that there's a space in between here that nothing is being placed, right? But we can see here that actually we have a lot of electrodes being placed here. Now that's just to, enable us to be able to also pick signals, even more electrical signals from the brain that could actually, to a long distance, or to tell us how what is going on within the brain of the patient. Also, we can see here we have the A1 and A2, which are reference electrodes, which tells us practically that they really don't take part in the entirety of the procedure, but they're reference electrodes, we try to say that we try to make them as reference to in respect to other electrodes and other readings we get from the EEG. So I mean, it's a lot, but I feel like this little diagram could also explain a lot. You could actually decide to go read a bit more about it, and tells us how much, gives you more information about the EEG, the electrode placement of the EEG, and how it's being placed usually. And of course, yeah, we have a picture representation here also. You could see a picture representation of what I'm trying to say. We can see here from the picture we have. Okay, so this would be me, but of course, you know, we have a lady there. But in real life, this would be me, and this would be the patient, right? Coming for the procedure. You could see here from the little view we have of the machine, you can see the electrodes here all being attached to the board of the machine. This you can see here, and you can see that the electrodes are placed on the scalp of the patient, which is actually what you can see here. We have a very happy patient there, which is very good for us. So you can see it kind of takes a little bit more, a lot of electrodes here. If you could count, it would kind of look similar to what I have here to say. So that's just let us appreciate even more what the EEG does and what it's all about. Because of time going forward, yeah, I like to talk about, of course, the nerve conduction study. So basically, a nerve conduction study is also an advanced neurodiagnostic test that evaluates the function of your peripheral nerves. A nerve conduction study can help detect the presence of an extent of peripheral nerve damage. Your peripheral nerves are the nerves that lie outside your brain and the spinal cord. So more specifically, the nerve conduction study checks the motor nerves, which from here you can see that the nerves that control your muscles and your movements, and the sensory nerves. These are nerves that tell you about control or carry signals about your touch, the taste, smell, and of course your sight. So to a certain degree, if you begin to have problems or complications, being able to move around, move your hands, move your legs, or move any part of your body, unlike how you used to, or you have an issue with sensory nerves, such as you lose your touch for your sense of touch, you lose the sense of taste, also smell, and sight, right? You could also, a nerve conduction study will probably be one of the neurological procedures you're asked to do, just to ascertain what's the level of damage that might be going on with the particular nerves that are actually responsible for those reactions. In this case, we are talking about the motor nerves and of course the sensory nerves. Of course, just for us to talk a bit more about what peripheral neuropathy we'll talk about, because we're talking about peripheral nerve damage here. So peripheral neuropathy would be happens when your peripheral nerves are damaged, and when your peripheral nerves are damaged, you're talking about a damage to your motor nerves. You already know what that is for, and your sensory nerves, and that's what a peripheral neuropathy will talk about. And most times that's what the reason why an NCS procedure will probably be requested for is will be as a result of a damage to your peripheral nerves. And of course, you could see there are some common problems, common causes of that would be diabetes, vitamin deficiencies, thyroid, alcohol use disorder, the infections, HIV, medications such as chemotherapy. You could also, as a result of a direct injury to your nerves, maybe as a result of trauma or a surgery that's kind of came back with a complication. And I mean, that's basically what the nerve conduction study and the EEG are all about, basically. All right, so we also have I have a picture representation of what a nerve conduction study looks like, right? You can see here from the first picture, you can see we're trying to, now this person probably has a damage with one of the peripheral nerves. If of the hand there, you can see maybe it could be as a result of movement or touch. Now what we do here is to try and stimulate. Now this, we call this this stimulating machine. This is a stimulator, right? And we have what we call your ground electrodes here. You can see here, and we also have the electrodes that conduct, the stimulation. So we have the positive, you can see the red, and we have the negative also, where you can see the black there. And this comes together to give us a projection of or a reply. Now we actually stimulate this part by sending in what we call shock waves or through that particular site, and that would cause an involuntary movement in the parts of the part you're trying to stimulate. In this case now is the hands, right? So if we stimulate at a particular energy level, you can see the two handles here, the 2 pointers here, that we stimulate at this point, and we stimulate it. And when we stimulate it, we send shock waves through this the patient. Now the shock waves that we send through the patients is what is being conducted by the nerves and the muscles, right? And the conduction from that nerves and muscles gives us a signal, a reply, like a reply, and that reply tells us the level of severity of nerve damage, nerve damage to that particular part we are testing. So you can see here, once that is stimulated, the shock waves goes through the patient, the reaction comes back, and we have like a screen. Yeah, we do have a screen that tells us we have like a shock waves that comes out the screen that tells us, okay, this is the level of severity going on within this patient. Now, so you can also see here, because like I said, we have to, it could be any part of the body, right? It could be your legs, it could be the hands, it could be any parts. So you can see here also, we have a representation of a patient a nerve conduction study being conducted on a patient on the peroneal nerves at the lower limb, peroneal, which is also the legs here. You can see also here that we actually have some sort of a different brand of nerve conduction studies equipment being used here, not looking like this one. That tells us also that we have a variety of nerve conduction study machines that comes in different ways, but of course they carry out the same function, right? You can see here also, the stimulating points here being carried out, the point of stimulation here. You see the positive and the negative being put together. So of course the patient is stimulated and the shock waves are sent through. The conducting electrode receive and give us back a signal after probably causing an involuntary movement of that particular part, in this case, the leg also. So we move forward. Of course, we try to talk more about, try to see more, examples of what a nerve conduction looks like. You could also see here, we call this a tibial motor nerve conduction study. You can see we're trying to also stimulate here. You can see the stimulation, the stimulating being done at this point. You can see the ground electrode here, and you can see the receiving electrodes here. The black and you can also see that it's a very different type of equipment, also different from the ones we just saw, but they still all carry out the same function, right? You can see the red and black machine equipment there. So when we stimulate, we also get the same stuff. We also have what we call a radial motor, still checking different parts of the body and how the signals are being sent back. They also look quite similar to this one. So they all take the same function. They do the same thing basically. Now I would like to just talk about, in terms of memory disorder, Alzheimer's disease and how it relates to the EEG and the NCS study. So we all know that it's a common disease, another common form of dementia that affects a lot of people, mostly people of the older age, 65 above, but we could also have it happen to people below that age, of course. We could have early onset of such disease. Yeah, so basically it's to goes around, we're talking around the effects of Alzheimer's disease, dementia, what it does to you, and what happens, right? Here we have, according to the National Institute of Neurological Disorder, so stroke. Alzheimer's disease is a progressive, non-degenerative disease that occurs when nerve cells in the brain die. Disease also results in the following: impaired memory, thinking, and behavior. It causes confusion, restlessness, of course, personality and behavioral disorders. We have impaired judgment, impaired communication. These are all the effects of, if any form of the dementia that you probably would have. And yeah, we have a picture representation also here of how a brain with dementia looks like. You could see here a normal brain, and a brain that has been affected by a disease or a kind of memory disorder. In this case, Alzheimer's disease. You can see the contrast and the change in look and in structure of the brain there. That's the effect, right? Of course. So it's been a very much new neurological disorder that's still been under study. Still, people are still trying to understand more about the memory disorder. It's not really an awesome knowledge yet. So we have a lot of things like science is trying to fully understand what causes the disease, although age, personal health, family history, and genetics, and all those are believed to contribute. But of course, we can't say certainly for sure that those are the major reasons why we have memory disorders happening. We're still under study. We're still trying. Research is still being done to have a better understanding of what memory disorder looks like. Of course, a recent publication by Professor Eli S. Neiman on electroencephalography and dementia and encephalopathy actually talks about the relationship of between EEG and dementia and how it works. We have another research that have been carried out in China, within the aging and elderly groups, and being done by Xin Yang and Lu Yu in 2022. They did a sample size of 74 patients with mild cognitive impairment, and 21 of them had Alzheimer's disease, and 82 healthy elderly individuals also. All participants underwent a peripheral nerve conduction, which is a nerve conduction study, and neurophysiological evaluation. And of course, this was used to assess the nerve conduction in the limbs, and comparisons were made between the two cognitive groups. Furthermore, the relationship between peripheral nerve conduction study and cognitive function was further investigated. So, like we said, this is a disease that is still being studied, that we're still trying to understand a few more about it. So definitely, we're not going to say, it's not going to be like, we're not saying EEG or nerve conduction study gives us the final outlook of how to properly diagnose memory disorder or such as dementia. Of course, we still have a couple of other procedures still has to be carried out, in which everything will be clinically correlated. Sometimes we have to carry out the mental status test, the blood test, the lumbar puncture also has to be checked. The analysis and checks also has to be done. Sometimes we have to do the CT scan and also the MRI. And also a lot of, just like my colleague, the physiotherapist, has said, a lot of physiotherapy sessions also come into play. I mean, entirely the treatment for an Alzheimer's patient is more collaborative than it being singular. So it's not really sectionalized to one particular for particular tests or particular departments or particular clinical procedure or diagnostics. It's all more or less like a wholesome treatment for a particular patient that might have this disease. Yeah, you can see here, just trying to furthermore try and relate what's the EEG to the memory disorder disease. You can see here we have a typical EEG representation of what a form of diagnosis that was made from an EEG for Alzheimer's disease. You can see here we have the parameters like I showed earlier in the diagram, the placements, and you can see here this is the region strip of an EEG. You can actually see here that every region strip is a representation of a particular point of that the electrode has been placed. Like I pointed out, you could actually see here. I know it's a little bit blurry, but you could see here the FP1, FP2, like I talked about, the F3, F2, F4, A1, C, the T3, the P2, and O1, O2 are all particular positions where the electrodes for the EEG has been placed. And we can actually see the reading strip or the reading coming out of the reading strip here, telling us what particularly is going on within the brain of that particular patient at that particular point. So I mean, this is just trying to relate it a bit to what we're talking about, right? I mean, this would ordinarily give us an idea of what's going on with the patient, but actually help us to also determine what other diagnostic procedures we could try to do to get a more wholesome outlook of what's probably going on. As a new diagnostic highlight professional, this is very important to me because this, to a large extent, tells me if what is being done can be correlated to other tests that has been done in respective of that particular patient, and if it can be correlated, if it's in line with what we're trying to achieve. So it's important that the EEG is done effectively and efficiently to give us a very, very nice, very good reading, an authentic reading that we can actually rely on and use to correlate with other diagnostic procedures that might have been done. Furthermore, we also have diagnostic representations of what the EEG looks like. Yeah, when we are talking about, like, this is a typical EEG in dementia also. You could also see here that we have readings from different points of the brain where the electrode has already been placed, and those readings are telling us a few things that we need to know about what's going on within the brain of the patient, and of course the wave signals that come out here and the amplitude at which they come out, the speed at which they are coming out tells us a lot about the situation of the brain of that particular patient, and also to a large extent enable us to be able to relate what we have on the EEG to other tests that might have been done, like I earlier stated, might have been done for the patients also, to ascertain in the total view what exactly is going on, and if we can correlate or we can diagnose this to be a memory disorder of some sort. So this is like a picture representation also. You can see how it is quite similar to what we have here also. Right. So we move now to this. This is a nerve conduction study. Practically, what's on the screen, right? What it looks like. Now, like I earlier showed us, when we stimulate a patient from the picture earlier showed, and we send the shock waves through the patient, and the shock waves, of course, we get a signal, a return signal from the nerves and muscles of the patient. Now, this is what it looks like on the screen, right? This is what it looks like on the screen. You have the signal coming back in this format, and differently looking. They look different And that difference tells us a lot about how we have what we call a latency. We talk about what we call the conduction velocity now, and the amplitude also. These are the major parameters that we look at the nerve conduction study. Now, the level of conduction velocity, the level of latency, and the level of amplitude signals that we get back from stimulating the patient tells us to a large extent what the level of peripheral damage, nerve damage to that patient, and which will also give us an idea to a large extent, the disease or what is actually going on with the patient. So this is how, the feedback, like I said, is the signal feedback looks like. You can actually see here from, the representation here, where this particular signal came back from the testing of the wrists and the elbow of the patient. That would, now the wrists would look like this. Sorry, I have to go back a little bit there. Now this is retesting the wrist of the patient. So what we're seeing here, this is the picture here. Now the feedback we are getting there is what we're seeing here. Now this is the feedback of what we just saw. Now, from the wrist, the wrist part, you can see here that we have a value for the latency. So we had the value for the duration, the amplitude of, of the test, and also we come here to talk about the conduction velocity. We have the distance that is going to be measured, between the place of stimulation. And though I have to go a bit more deeper into this for us to really understand, but there's really no much time for us to do that now. But basically, just trying to give us an awesome view for us to further appreciate what the nerve conduction study is all about, and how it helps in diagnosing memory disorders as far as in relation to nerve conduction study and all. And you can see here, similarly, we also have, this is more like a template of what it looks like. This tells us a bit more about the extent of what the nerve conduction study is able to do, and the extent to which the parts that has been able to be tested. We have a peroneal nerve here. Like I said, we're trying to, nerve damage tells us it's a peripheral nerve damage. So we have different nerves we can actually check. We have the peroneal nerves here. We have the tibial, we have the sural, we have the median nerve, we have the ulnar, right? And we have different stimulation sites where we check, where we stimulate, for the peroneal, for example, here, you stimulate the ankle, you stimulate the fibula, you stimulate the popliteal fossa. And when you do that, these are points on the patient, on the body that you actually, you test just to see the nerve reaction there, just to see the muscles response, for us to know, the level of damage or level of nerve damage severity, to be able to relate to other things. And you can also see we have the amplitude, the latency, and the conduction velocity here, which are more or less like the major parameters we look out for when it comes to nerve conduction study. Now, of course, we have a larger template of values that tells us that, that gives us an idea of what a normal value should look like and what the obtained value is. Now, the obtained value, in comparison to the normal value, tells us that the level of damage, or whether there's a lot of damage going on, or whether there's a mild damage, or whether it's actually even normal, right? Whether there's nothing actually going on, right? So we have to take those values and you relate it to normal values, which universally there are a lot of values you can actually extract universally and use for yourself, and you have that to give you your response of what it looks like, of what it tells you the level of severity, right? Of course. So basically, these are the parts of the body that can act, that the nerve conduction we can test. We have the peroneal, which is the lower limb part. The tibia is also lower limb. The median is the upper limb. The ulnar is upper limb. So basically, for nerve conduction studies, we check the upper limbs, check the lower limbs. That's the upper and the lower parts of the body. Depending on what the complaint is from, if the patient is talking about difficulty moving at the upper limbs, or it's talking about he or she having difficulty moving or having sensation at the lower limbs, it tells you what parts of the nerves you have to check to to ascertain what's going on, whether through the upper limbs or at the lower limbs. Yeah. So I have the full PowerPoint presentation for you people that are more interested to actually look a bit further. All right. So now we're talking about the treatments. Of course, like there right now isn't any specific kind of the treatment for memory disorders. As like I said, is something that is still being researched upon. A lot of investigations are still going on concerning memory disorders and how we can be treated. So a lot of things are still being discovered day in, day out. So I mean, I'm hoping that with time we'll have a better wholesome and total approach to treatment of Alzheimer's disease or any form of memory disorder. But of course, what is being done currently, just like the physiotherapies has also explained clearly, it's a whole lot of collaboration or collaboration between treatments, between whether clinicals or diagnostics, to give a patient that has a memory disorder a wholesome treatment to that increases the possibility of the patient coming back to being okay or being in top health again. So a lot of things are going to be being put together. To a lot of tests that are being done, have to be done, and everybody has to come together to try and help out and contribute their quota in terms of diagnostics or clinicals to further help a patient that has a memory disorder to come back to full health. All right. So in my conclusion, just to try and relate everything together, Professor Eli S. Niemann concluded that from his research, a computer analysis of the electroencephalogram may help reveal subtle changes in ischemia disease, right? So EEG may differentiate between a generalized or focal abnormality. And when you're talking about generalized, you're talking about the abnormality happening from every part of the brain. And when it's focal, we're talking about it actually coming from a particular part of the brain. And the electrodes, like I said, the electrode placement of the EEG helps us to be able to understand, looking at from the output or the reading strip, tells us what particular area or what particular points of the brain, whether the what's going on is generalized. If it's generalized, we practically see the symptoms or the wave signals popping up everywhere. But if it's focal, then we probably will see the wave signals popping up, the wave abnormality signals, the wave abnormalities, per se, popping up at specific points at the brain, specific points of the brain, which you are going to be able to highlight, tracing the reading strip to the electrode placement. So it could be from the temporal point, it could be from the temporal electrode. The wave abnormality could originate from the parietal, the occipital region. So that enables you to be able to sectionalize the abnormality coming up. However, of course, the accuracy of diagnosing memory disorders through encephalography and nerve conduction studies alone, like I said, may not be ideal, suggesting whether neurodiagnostic processes alone can be used as a marker for a diagnosis of memory disorders. This, however, provides new opportunities and possibilities for future research. Now, looking at what Prof. Niemann did with the EEG, he has been able to tell us that, of course, yes, the EEG can help us to, to review subtle changes in the disease, looking at the points of origination, whether it is focal or generalized. And of course, we have to study the waves carefully to tell us that, okay, this the change in this wave, what does it tell us? What is it trying to say? Can we correlate it with other diagnostic procedures? And he has also further gone to, to highlight that, of course, we cannot just base treatment for memory disorders, like I've stated earlier, on just the EEG or the EEG test alone, right? Because we also have to put other diagnostic procedures into consideration. We have to correlate it together, which what I've said, which emphasizes what I've just said, that it is a collaborative effort when it comes to memory disorder treatment in patients. Is much less like a collaborative effort. But of course, every single diagnostic procedure has their own particular contribution, and of course, the EEG has a large contribution because, of course, we are talking about memory disorders, we're talking about the brain, and EEGs basically is one of the advanced brain neurological diagnosis procedures that actually tells you, to a closer degree what goes on in the brain. Now, all right, so to also try and relate another research done in China by Xin-Yin Qian, I hope I got that pronunciation correctly. He also concluded that peripheral motor nerve velocity may correlate with early cognitive impairment in Alzheimer's disease. Now, of course, we are talking about nerve conduction study now. Qian is saying from his research that he conducted that, we can actually relate Alzheimer's disease to a loss or a peripheral motor nerve velocity change or damage. We could actually relate that to Alzheimer's disease. So which tells us that, of course, the nerve conduction study also, despite the fact that it's an advanced procedure which currently is not, probably widespread for example, in Nigeria, has a lot it can offer for as far as memory disorder is concerned. It tells us that, if you do a nerve conduction study, for example, and you have a kind of there's a kind of change, or a kind of the values or the waves you're getting from your the motor response is kind of reduced, or it's below the normal range. That could also be, an early signal of an early cognitive impairment. We should tell you that we can also relate easily to other, to other tests that you're done, and you can actually relate it to the other particular tests and correlate it to see if the reduction in the motor nerve velocity of that patient could have anything to do with the fact that he has a memory disorder, or and that could also help. And however, though, Kian also stated clearly that the accuracy of different cognitive qualifications and the value of early diagnosis are not ideal when the peripheral motor velocity is used alone. So whether peripheral nerve function can be used as a marker for early diagnosis of AD still needs further clarification, but provides a new possibility for future biomarker research. So what Prof. Niemann and Kian are actually saying here from the neurological research and study is that yes, the EEG and nerve conduction study can contribute a lot and immensely to us trying to diagnose a patient for memory disorder, or to us trying to clarify or try to correlate a result from EEG or the nerve conduction study to other diagnostic clinical investigations that have been done, for us to see if there's a wholesome correlation to what the patient is having. And that's basically what Professor Niemann and Kian are trying to say here, while highlighting clearly that of course we cannot, because we're still trying, it's still something that is being going under research, it's still being investigated, that we cannot totally rely on just the reports, the EEG results, or the EEG procedure and the nerve conduction procedure to fully diagnose a patient that has a memory disorder, dementia, Alzheimer's disease, or whatever the case might be. We should further, highlight that Other diagnostic procedures also has to be taken into consideration. Other clinical investigation also has to be taken into consideration alongside the EEG and the nerve conduction study. All right. Thank you very much. Thank you.
So it was the talk by, I'm sorry, there is a voice disturbance. I'm sorry. Okay. So it was the talk by Dr. Abolaji Arinfolame. And thank you so much, Dr. Arinfolame, for explaining the EEG nerve conduction study and the relation with memory disorders. All right. So
Yeah.
anyone? Okay. So everyone who are having questions can come up. As it was detailed insights. I repeat, if you are having any questions or anything you would like to add on, then please come up. You can ask your questions in the chat box or simply ask the question by unmuting yourself. Till then, I would like to mention that we will be needing your designation with your full name and your email address for the certificates formations. So please share your designation as it is Miss, Mr., Doctor, Professor, and your full name and your email address. Please share the same in the chat box. And this is for all the attendees, as we are already having the details of all of our speakers with us. I repeat, if you are having any questions, not only from Dr. Arinfolame, but from all 5 of our speakers, please come up and ask your questions. And yes, it was the last talk of the day, as unfortunately we couldn't have Dr. Agrawal with us today, as he had to go for a sudden condition. All right. I repeat, please do not forget, dear attendees, please do not forget to share your designation, your full name, and your email address with us. Also, let me share about the next event we are going to have, which is on 16th of June. Here is the direct link for the event, and it's all about brain inventions and discoveries. The latest discoveries and inventions on brain. And we are already having a speaker, our well-known Dr. Sampurnam Webster, and she will be presenting about avatar therapy in treating auditory hallucination. If you are excited to know about avatar therapy, please do join us. Yes, Yes, dear Amri, yeah, we will share the certificates on the email you just shared with us. Please make sure there is no spelling mistakes. There is your full name. Please recheck, so there won't be any mistake from our side also. You all will receive your certificates by the next 3 working days. Okay, Mr. Anuj, which PPTs are we talking about? I mean, any particular speakers or the introductory PPTs? Okay, you will definitely get the introductory PPTs in our discussion portal for this event. And if you want the PPTs from any particular talk from any particular speaker, then we have to take permission from the speaker itself, and only then we can share the PPTs with you. Otherwise, we are having our YouTube channel, and all of our talks here are recorded, and we will provide the videos there on our YouTube channel. You can go there and definitely watch today's talks as well as the earlier ones also. First, let me share the direct link for discussion portal for memory disorders and conditions. Okay, here is it. I repeat, please call me Ghazal. As I feel it easier to interact with all of you, and it's more comfortable. So if there are any more questions, from any of our speakers, please come up with your questions. Also, our today's event was live streamed on LinkedIn, and it was just a trial. So thank you so much for bearing with us. We will definitely try our best to live stream our events on LinkedIn and YouTube also in future. And also we request our speakers to please share their recorded video presentations in case they are not 100% certain that they will be able to join the meeting. It will help our attendees to get a hold on your talks, on your presentations. Yeah. So I hope we already have all the details, all the names and emails in our chat box. Please connect yourself with us on our social media platforms and subscribe on the website also to stay updated about our every upcoming activity. Indeed, thank you so much to everyone for joining us today and your contributions to the event. Thanks to Mr. Emre also. All right, if you need any particular information, please write down in the chat and we will definitely reply. So this was it for today. Thanks to all of our speakers, all the 5 speakers of the day, and all the attendees also. Thank you, Dr. Nakieta Lankster and Dr. Tshetiz Dahal and Dr. Amara Rafiq, Dr. Abolaji Erin Polame, and Dr. Ayodeji Ayomiko. Thank you for providing us your precious time, and a special thanks to Dr. Sampurnam also for coming up as a support and sharing her knowledge about memory disorders in the introductory section. Thank you, and hope to see you again in our future events. All right, this was it. Thank you. My name is Ghazal Johar and Namaste from India. And we will definitely meet in our next events. If you are interested to be a speaker, please do not forget to share your research abstracts with us. I hope you already, you all are having our website address also. If not, I'm sharing it here. Please do visit us and find the events that suits your interests and join us for the events. Thank you. Thank you and take care, everyone. See you again. Bye-bye.